Mary Hagedorn, and I'm a marine biologist at the Smithsonian Institution and the Hawaii Institute of Marine Biology. My assistant Jeannie Carter and I have been working on coral cryopreservation and conservation for the past 9 years. Coral reefs are keystone ecosystems in our oceans. Coral are animals. Some people think they're rocks, some people think they're plants, but they're really an animal, and they reproduce by producing sperm and eggs. And without them, we could potentially lose many of the animals that now either live on a reef or depend upon a reef for their livelihood. The reason that this reef conservation is so important is that coral reefs around the world are declining at a rapid rate. We do not know what the future will bring, but I would like the next generation to experience the beauty and wonder of a tropical coral reef. And it is not okay for me to watch coral reefs go extinct while we still have the power to change that future. We have perfected the ability to cryopreserve sperm, coral sperm, and coral embryonic cells.
When we go somewhere to do our field work, if you can just imagine what might go into a human fertility clinic, you're going to have folks that need to check the sperm health, and they'll need microscopes for doing that. You'll need the freezing equipment, and of course the containers for keeping the samples frozen. All those things that you could see in a big human fertility clinic, we are setting up at sites in the field, creating a coral fertility clinic.
The most complicated part is making sure that we're there at the right time and we have good weather. That's really the most complicated part.
We have the advantage of being at the Hawaii Institute of Marine Biology, which is actually out on the reef.
For the past 2 summers, Australian summers, we've gone to the Australian Institute of Marine Science to cryopreserve and form a bank for the Great Barrier Reef. The Great Barrier Reef holds much of the genetic diversity, coral genetic diversity in the world, and it brings in $6 billion annually to the Australian economy.
When we're in the field, our corals spawn at specific times at night, several days after the full moon. So depending on which species we're working on and how many species, we'll have between 2 and 6 nights, almost a week, of staying up, waiting for coral to spawn, and then processing sperm, processing embryonic cells. There will be several of us, 4 to 5 people, around tanks with the coral inside. We have red lights on because the coral are sensitive to the light, and that's actually one of the cues that they use for spawning, the moonlight. Each coral is kept separate from the other coral because we don't want them to fertilize themselves until we need them to be fertilized. So we'll collect eggs and sperm, or in the case of many of our corals, bundles, which have eggs and sperm inside of them, which we'll break apart. So we'll be collecting for anywhere from 15 minutes to an hour or so, and then we'll take everything back into the lab, and then we'll start assessing the sperm on the microscopes. So we'll look at sperm motility, we'll look at sperm density, how much sperm do we have, is each coral healthy, are the sperm swimming well, and then we'll choose the ones that we want to keep for our bank. At that point, we'll then put the sperm into cryoprotected, which helps the sperm be frozen, and then we'll go through our cryopreservation process. After the cryopreservation process, we'll thaw a few samples and make sure that everything looks okay, and then those sperm samples will go into our bank. As some of us are freezing, some other folks in our group will make coral larvae, and then those coral larvae will be used hours later to freeze coral embryonic cells. So we'll take the embryos, the larvae that are there swimming, fertilized and alive and happy, and then we'll use a glass homogenizer to break those larvae apart. This will break them apart, but the cells will not die. So we have live coral cells in solution, and then those will get frozen similarly to the sperm, but at a different rate. The coral embryonic cells are taken at points in their development in which each cell in that embryo is pluripotent. This means that those cells can grow into whole adult corals. So if you imagine the animal starts out at 1 cell, 2 cell, 4 cells, those cells have to turn into the whole animal. So those will be frozen in our bank, and then at some point someone can thaw them out and grow whole corals. So So the process of doing the embryonic cells is a little interesting because we have to wait until they're at the point in time that we want to take them. Sometimes this means we're staying up until 5 or 6 a.m. for various time points and then getting to go ahead and do it all over again the next night. During our visit to the Australian Institute of Marine Science in November of 2012, the reef recovery team had great success. We were able to add 3 new species of coral to our bank, giving us 5 species total. For Acropora tenuis, we now have 228 billion sperm cells. For Acropora mellipora, we have 14 billion sperm cells and 1,440,000,000 embryonic cells. For Acropora loripes, we have 22 billion sperm and 720 million embryonic cells. For Platygyra dahlia, we have 1 billion sperm cells and 720 million embryonic cells. And finally, for Platygyra lamellina, we have 1,220,000,000 sperm cells.
One of the great successes of our trip to the Great Barrier Reef in 2012 was the beginning of a coral nursery. In collaboration with the Australian Institute of Marine Science and their CSIM facility, we now have small coral growing, and in this group we use both cryopreserved and fresh sperm. The two groups were similar in their ability to be fertilized, develop, settle, and take up zooxanthellae, which you see as brown dots. Both groups are growing and are doing quite well. What this means for us is that the cells that are now in our bank, it's proof that they will work in the future to create new coral and help diversify shrinking coral populations.
There's no question that the state of our planet is in peril.
We are really the only laboratory in the world that's doing this. We're losing genetic diversity in the oceans very fast. And with such a small group, we can't move fast enough to bang down all the diversity in the ocean. We need to train more people, we need more funds to be able to maintain ourselves and the people that we train, and we need to spread the word.